Hey everyone, welcome back to another session of Biology and Human Welfare. In today's class, we are going to discuss about the Entamoeba histolytica. Do you know the reason of death of famous Mughal Emperor Akbar? I'm sure you must be knowing about Akbar. But do you know the reason for death of the Akbar or most famous Telugu king Sri Krishna Devarya? Do you know about them? Or do you know about the death of them? It was because of the Entamoeba histolytica, right? Now, what is the meaning of Entamoeba histolytica? Let's figure it out. Entos means within. Amoebae, you remember amoeba? From that you can remember it. Amoebae means change. That means, amoeba does not have a fixed structure, right? So it changes its structure. Now, histos means we have already seen it means tissue, and lysis means to dissolve it. Now, one more thing, class, it's just a trick for you to remember. Enta means you can remember with the enteric cells. We have spoken about the enteric cells in our previous class. These are the cells of intestine, right? So where you will find Entamoeba histolytica, you are going to find them in your intestine, right? Now, what are the characters of Entamoeba histolytica? If we'll talk about the systematic position, what is the phylum? Protozoa. Now, class, we have studied about the 11 phylums. Among 11 phylums, have we studied about the protozoa? No, we haven't studied, right? So, ma'am, have you written wrong? No, I have not written wrong, class. We have to study 11 phylums, but apart from that, we have many other phylums as well, right? Now, subphylum is nothing but your Sarcomestigophora. Sarcomestigophora. Now, in comment box, write about the Mestigophora. In which chapter we have studied about the Mestigophora? Do you remember it? It has something to do with the flagella. Do write it in the comment box and let me know who remembers. Class is your Rhizopoda. Rhizopoda is nothing but the organism who has pseudopodia. Are these macroscopic? Yes. They are monogenetic. Now, whoever has studies yesterday video, they know that what is the meaning of monogenetic. Mono means one. So, monogenetic means organism completes its whole life cycle in one host. Mono means one, so it is going to complete its whole life cycle in one host. What is the target site? Target site is the large intestine. In large intestine also, which one? Colon. What is the cause? What does it causes? It causes amoeboid dysentery or amoebiasis. Put star mark over here. This is the neat question. Now, all the four, whatever we are studying, you're supposed to remember the microorganism name along with the what does it causes. Okay, you are supposed to remember it. Why? Because it comes in neat as an MCQ. Okay. Now, amoebic dysentery or amoebiasis is nothing but the diarrhea which is caused by amoeba. What is diarrhea, class? It is nothing but your loose motion. It is nothing but your loose motion. So, loose motion when it is caused by amoeba, we call it as amoebiasis. What is the mode of infection? It is through contaminated food and water. See, if the target site is large intestine, small intestine or alimentary canal, so it means that the mode of infection will be always contaminated food and water, right? Now, mechanical vector. Do you remember about the mechanical vector and biological vector we studied in the previous class? Mechanical vectors are the vectors which are the physical carrier. What about the biological development? No biological development occur over here. So, mechanical vector are the physical carrier. For example, for Entamoeba histolytica, your physical carrier or the mechanical vectors are housefly and cockroaches. They are cosmopolitan. Now, often you are going to use this term for bacteria, fungi, mostly for bacteria. Now, cosmopolitan is nothing but it is present everywhere. Write this meaning in your textbook. It is present everywhere. Most commonly, you will see them in tropical and subtropical region. Do you remember when we studied about biodiversity? In biodiversity, we studied that we will be having more biodiversity or more diversity in the tropical region because it is near to the equator, right? So, same thing will happen over here. Now, commonly you will see Entamoeba histolytica in rural area or densely populated area. Now, why only these area? Because they are not going to follow the hygiene properly. So, due to their poor hygiene condition, rural people and densely populated area will have Entamoeba histolytica. Now, first we are going to study about the structure of Entamoeba histolytica followed by the life cycle. Please listen very carefully and make all the notes. Once we are done with the stages, then we go with the life cycle. If we won't understand the stages, we won't be able to understand the life cycle. So, Entamoeba histolytica has three stages. First is trophozoite, second is pre-cystic, and third is your cystic stage. Right? When we talk about the trophozoite, it is the most active stage. Since it is most active, it is motile stage. And why it is most active and motile? For feeding, for food. So this stage of Entamoeba histolytica is going to feed on the organism or feed on the host in order to get the nutrition. So this is most active, motile, feeding stage, and at the same time it is pathogenic. How pathogenic? Suppose if somebody is feeding on you, so obviously it is pathogenic, no? It is harming your cell, correct? So it is pathogenic. Now what is the target site? We have already studied. It is the large intestine. But in large intestine also, which one is the target site? It is mucosa and submucosa layer. Now class, we have already studied in class that this is, if this is your gut, okay, now where I have written gut, it is nothing but your lumen, empty space. I'm trying to show you large intestine ka, okay? Now this empty space may, that is in lumen, you have undigested food or unabsorbed food will be present over here. Now the first layer which you can see, it is nothing but your mucosa. It is nothing but your mucosa. Then we have second layer over here. This second layer, we call it as submucosa. This second layer we call it as submucosa. Then we have third layer which we call it as muscularis, which you don't have to study now. And we have fourth layer, the outermost one, we call it as serosa. We call it as serosa. Right? Now, target site is your mucosa. So that means your first and second layer, mucosa and submucosa, are your target site for whom? For Entamoeba histolytica. Right? Now locomotion, how it is going to, it is a motile stage. So how it is going to move? By using the pseudopodia. How is the pseudopodia? It is single blunt finger-like structure. It is covered by plasmolemma. What is plasmolemma? It is nothing but the plasma membrane. It is nothing but the plasma membrane. Cytoplasm is differentiated into two. One is your ectoplasm, other one is your endoplasm. Now if you remember in amoeba, we have already studied about the cytoplasm. Now in amoeba, this outer cytoplasm we call it as ectoplasm. Inner one we call it as endoplasm. Same thing over here. The outer one we call it as, where is it? Outer one we call it as ectoplasm. Plus you need to draw this diagram. It is super important. Okay. So outer one we call it as ectoplasm, and inner one which you can see here we call it as endoplasm. Ectoplasm is the outer layer. Endoplasm is the inner layer. Ectoplasm, if you see, it is clear. Can you see this yellow color one? It's completely clear. There is no granule. That means as a chote dane. Okay. These granules are not present over here, and this is viscous. Viscous, like, you know, little thick it will be. Liquid bhi hoga, thick bhi hoga, like honey consistency it will be, right? Now, if we'll talk about the endoplasm, it is a fluid-like structure and it has granules. See this endoplasm, you have chote aise dane present hai na? These are called as granule, and it is also consisting of ribosome. Let me show you. This is your ribosome, and then you have food vacuoles. Pay more attention to food vacuole. This food vacuole, let me draw it with another color. Okay, see here, one food vacuole and then we have another food vacuole. If you will see, food vacuole has RBC, another food vacuole has bacteria. Why this has RBC or bacteria? Because it is feeding on the mucosa and submucosa layer. So their blood vessel will be there, blood vessel hai to RBC will be there, right? And then in intestine we have bacteria as well. So due to that, what they will have in their food vacuole? They will be having bacteria as well as the RBC, right? And vesicular means vesicle-like structure, this like structure. Their nucleus is something different and special. They have cartwheel-shaped nucleus. Now write in the comment box where we have studied about the cartwheel shape, cartwheel structure, where we have studied. Now, see here, this is your nucleus. This is your nucleus, which is cartwheel shape, and in middle they have tried to show nucleolus, but it's not necessary it will be present in middle. But for trophozoite, the structure is like that, so we are going to follow it like this. Okay, nucleolus is nothing but it helps in the formation of rRNA, which in results help in the formation of ribosome. Helps in the formation of ribosome, right? Now, food vacuole consists of your RBC, your bacteria, and fragments of epithelial tissue. Epithelial tissue is nothing but your surface tissue. More about it, we are going to study in the different class. Now, let's see this diagram class. You can see the elevated thing over here. This is nothing but your pseudopodia. वो थोड़ा सा नीचे आ जाएगा when we'll go in the next stage, right? Contractile vacuole, endoplasmic reticulum, Golgi, and mitochondria are absent over here. So, if mitochondria is absent, what changes you will see in the organism? Organism won't be able to produce organism won't be able to do aerobic respiration, because when mitochondria is present, it goes with the aerobic respiration. Without mitochondria, there won't be possibility to go with the aerobic respiration, so it becomes obligate anaerobe. What is obligate anaerobe? Obligate means compulsory, so they need absence of oxygen for the growth. Okay, growth of what? Growth of trophozoite stage. Now, it produces the proteolytic enzyme. What is the name of the enzyme? It is histolysin. When I say histolysin, histo means what, class? Tissue. So, what it is, lysin means break it. So, it is going to digest what? It is going to digest your tissue. It is going to digest tissue. Kiska tissue? It is going to digest the tissue of mucosa and submucosa. Where you will see mucosa and submucosa, you are going to see in the large intestine, right? So that's how it is, what it is going to do. Let's see here, releasing some amount of blood, because mucosa, submucosa के पास blood vessel भी होगा, tissue debris, and these all are ingested by the trophozoite. So if I'll ask you to find out whether this is trophozoite stage or other two stages, how will you figure it out? So the characteristic feature of trophozoite is nothing but the presence of RBC in the food vacuole, and the second was the cartwheel shaped nucleus. These two are the characteristic feature of the trophozoite stage. Let's jump on the next stage, that is pre-cystic stage. Pre-cystic stage is non-feeding. What was trophozoite? It was feeding stage. Pre-cystic is non-pathogenic. Trophozoite was pathogenic. It is non-motile, it was motile. Target site is lumen, जो आपकी intestine की खाली जगह है जहाँ पे आपका food present है. ठीक है, कौन सा food? Undigested food. Okay, undigested food is present. उसको हम क्या बोलते हैं? Lumen बोलते हैं. वहाँ पे target site क्या था? Mucosa and submucosa of large intestine. If you see the structure, it is small spherical shape. This is the correct structure. This is not the correct structure. Okay, this is the correct structure where you can see pseudopodia, but a little less it will be there as compared to the trophozoite, right? And then cytoplasm is going to store two things over here, that is your glycogen mass. You can draw it over here, or as per your textbook you can follow, and then you have chromatoid bars like this. Okay, chromatoids bars like this. So if I'll talk to you about glycogen, you know glycogen is the stored carbohydrate, and it is stored form of carbohydrate in whom? In animals. What is the stored form of carbohydrate in plants? Write it in the comment box. So it will be storing glycogen for the source of energy. Then what about chromatoid bars? It is nothing but it is made up of ribonucleoprotein. What is ribonucleoprotein? You must have heard about ribonucleic acid, that is for RNA. So same way, ribonucleic protein is nothing but RNA plus protein. Together we call it as ribonucleoprotein. These both are going to act as a reserve food. Okay, your glycogen and chromatoid bars are going to act as a reserve food. Now what is the need for them to act as a reserve food? Why? Because it is a non-feeding stage. अगर वो खाएगा ही नहीं तो वो जिंदा कैसे रहेगा? Obviously with the stored form of energy. So it has stored form of energy in it. That's what it is going to utilize for its survival. Then the last stage is your cystic stage, which this diagram is correct. You're supposed to draw it. Structure is round and spherical. Again, cytoplasmic storage is same as the pre-cystic stage. And now the change over here is it was a pre-cystic stage. That means before cystic. Cyst हम किसके लिए use करते हैं? Do you remember we studied about if we want to prevent it from any unfavorable condition, for example, desiccation. Right? Desiccation is nothing but your drying out. Desiccation is nothing but your drying out. Right? So surrounded by the cyst wall. When an organism, here this one you can see, this is nothing but your cyst wall. When an organism is surrounded by the cyst wall, we call it as it had entered into the cystic stage. What is cyst wall? It is thin, delicate and resistant. Resistant to the unfavorable condition. Resistant to unfavorable condition. Now, next process in cystic cycle, the very important process is nothing but your encystation. What is encystation? So cyst stage is nothing but the formation of cyst wall. This process of formation of cyst wall is nothing but encystation. The process of formation of cyst wall is nothing but your encystation. Now why we are supposed to do that? We have already studied for to tide over the unfavorable condition. Now when we talk about Entamoeba histolytica, what is the unfavorable condition for them? Now until and unless it is passed to the new host, it is in the unfavorable condition. That means जब तक वो host की body में अंदर नहीं रहता है, बाहर रह रहा है, it is the unfavorable condition for it. So what it will do when it is outside the host body, it is going to grow a wall around itself that we call it as cyst wall. That we call it as cyst wall, right? Now, soon after encystation, nucleus will undergo two successive series of mitotic division. So, this nucleus, after the formation of cyst wall, this nucleus undergo first mitosis and then form two nucleus. Then this undergoes second mitosis and two nucleus are going to form four nucleus, and this stage we call it as tetra, means four. Nucleus means nucleate. Tetranucleate cyst, or we also call it as mature cyst, and this is going to be the infective stage of Entamoeba histolytica, right? So what is the infective stage of Entamoeba histolytica? It is the tetranucleate cyst. Now, I hope all the three stages must be clear to you. So, we studied Trophozoite stage, which is a feeding stage, and then we studied pre-cystic stage, and then we studied cystic stage. Now, let's just enter into your life cycle. Please focus all of you and write these notes. Now, life cycle of Entamoeba histolytica, as we have started stages with Trophozoite, we'll start life cycle with the Trophozoite. So, Trophozoite undergo binary fission in the wall of large intestine. Wall means I'm talking about mucosa and submucosa. So these trophozoites, see, I'm not saying that ट्रोफोजोइट की स्टेज में वो ह्यूमन में एंटर करता है, बिकॉज वी स्टडी दैट इन्फेक्टिव स्टेज इज टेट्रान्यूक्लियट सिस्ट. सो कौन सी स्टेज में एंटर करेगा? इट इज गोइंग टू एंटर इनटू टेट्रान्यूक्लियट सिस्ट फॉर्म में. लेकिन क्योंकि हमने स्टेजेस ट्रोफोजोइट से पढ़ा है, हम लाइफ साइकिल भी किससे पढ़ रहे हैं? ट्रोफोजोइट से पढ़ेंगे. सो ट्रोफोजोइट विच इज द फीडिंग स्टेज. फीडिंग स्टेज में व्हाट इट विल डू? इट विल अंडरगो बाइनरी fission. Where? In the wall of the large intestine. Due to binary fission, it will produce large number of daughter Entamoeba. Now these daughter Entamoeba, obviously if they have to survive, they will be feeding upon whom? The bacteria which is present in the large intestine, and also the tissue which are present in the large intestine. Which tissue? Mucosa and submucosa. Now ultimately they grow in size and repeatedly divide again by binary fission. So what did we study? Trophozoite, binary fission, form daughter cell. Again daughter cell is going to feed. Again it goes for the binary fission and produce daughter cell. These daughter cell again feed. So this cycle keeps on repeating itself. After that, what happened? After repeated binary fission, that means you have lot many number of daughter trophozoites, right? When trophozoites increase in number, some of the trophozoite, not all the trophozoite, some of the trophozoite enter into the lumen of the large intestine. So अब तक कहाँ हो रहा था ये सब कुछ? अगर ये आपका गट का ल्यूमेन है, ल्यूमेन मतलब खाली जगह। तो अब तक वॉल में सब कुछ हो रहा था, ट्रोफोजोइट वाज फीडिंग, नाउ इट इज गोइंग टू एंटर इनटू द ल्यूमेन। ल्यूमेन में एंटर करने के लिए क्या हो जाता है, इट बिकम्स द प्रीसिस्टिक स्टेज। नाउ ल्यूमेन में ही ये स्टेज अगर आपका सिस्ट वॉल बना लेता है तो हम क्या बोल देते हैं, इट एंटर्स इनटू cystic stage, it enters into cystic stage, right? Now in cystic stage what will happen, ये हम पहले पढ़ चुके हैं, cystic phase में nucleus will undergo two mitotic division in order to form your tetranucleate cyst, right? Now these tetranucleate cysts are infective stage. These tetranucleate cyst is going to come out along with the fecal matter and it will be alive for 10 days. And other point which we can notice is these tetranucleus are basically going to be the infective stage. So यही है which are going to enter into our body as the infective agent, right? Now these tetranucleus cyst is going to enter into, mind it class, trophozoite is not entering in our body. We start with trophozoite just because हमने stages वैसे पढ़े थे, right? Infective stage is nothing but your tetranucleus cyst. Now these tetranucleus cyst reaches to the new host. How? Through contaminated food and water. अब तक क्या पढ़ा, ट्रोफोजोइट पढ़ा, अंडरगो बाइनरी फिजन के बाद वो फीड करता है, फीड करने के बाद बाइनरी फिजन के बाद डॉटर सेल बन जाता है, ये डॉटर सेल फीड करते हैं, फीड करने के बाद वापस बाइनरी फिजन से, ऐसे बहुत सारे बाइनरी फिजन होने के बाद आपका क्या हो जाता है, बहुत सारे डॉटर ट्रोफोजोइट्स बन जाते हैं, राइट? दीज ट्रोफोजोइट व्हाट विल हैपन, इट विल एंटर इनटू द ल्यूमेन एंड गोज फॉर प्रीसिस्टिक स्टेज, देन सिस्ट वॉल बना लेते हैं, गोज फॉर सिस्टिक स्टेज एंड देन न्यूक्लियस में 2 टाइम न्यूक्लियस की माइटोटिक division होती है, which results into tetranucleus cyst, which is the infective stage. Now it enters into the new host through contaminated food and water. They pass into small intestine of the new host. अगर contaminated food and water से आया तो obviously आपका किस तरीके से जाएगा, elementary canal की तरफ से जाएगा, right? तो वहाँ पे कहाँ पहुँचेगा, small intestine में. Now there the cyst wall, minded class, हमारे stomach में HCl होता है, that HCl is ineffective on the cyst wall, okay? Ineffective on the cyst wall. That cyst wall is going to get ruptured where, in the small intestine, by the action of enzyme trypsin. Now write in the comment box from where you get trypsin. Trypsin is released from question mark. Okay, now this what will happen, C class, again I am repeating, tetranucleate cyst था, उसके अराउंड हमने क्या किया था, cyst wall कर दी थी, right? अगेन बना रही हूँ यहाँ पे, ये देखो यहाँ पे 4 न्यूक्लियस थे और उसके अराउंड हमने cyst wall बना ली थी. So now you only tell me when this cyst wall will be broken down. This is the cyst wall. When this cyst wall, not this outer one is the wall, okay? When outer wall is broken down, what will come out? This tetranucleate amoeba is going to come out, and this process we call it as Excystation. पहले क्या पढ़ा था, Excystation. Excystation. Okay, Excystation is nothing but development of the cyst wall. Excystment क्या है? Excystment is nothing but the removal or the breaking of the cyst wall. Now after excystment, tetranucleate amoeba we call it as metacystic. Okay, metacyst. तो जैसे ही वो बाहर आया ना, उसको हमने दूसरा नाम दे दिया, जैसे आपका घर का नाम कुछ और होता है, स्कूल का नाम कुछ और होता है, तो now this tetranucleate cyst we are not going to call it as a tetranucleate cyst. The another name of tetranucleate is going to be metacyst, right? The 4 nuclei of metacyst undergo mitotic division. So 4 से कितना बनेगा? You are going to have 8 nuclei. You are going to have 8 nuclei. Each nucleus get a bit of cytoplasm. यही पढ़ा था क्लास हमने, multiple fission में कैसा होता है, multiple karyokinesis होती है and then followed by cytokinesis. Each karyokinesis is not going to followed by the cytokinesis. Each karyokinesis is not going to followed by the cytokinesis. तो क्या होता है यहाँ पे, multiple karyokinesis से cytokinesis follow होता है. तो हमारे पास 4 न्यूक्लियस ऑलरेडी थे, 4 न्यूक्लियस से कितना न्यूक्लियस बन गया, 8 न्यूक्लियस. नाउ ये सारे न्यूक्लियस के अराउंड साइटोप्लाज्म आ जाएगा and it is going to form the daughter Entamoeba. It is going to form the daughter Entamoeba, जिसको हमने दूसरा नाम क्या दिया हुआ, मेटासिस्टिक ट्रोफोजोइट. अभी देखो यहाँ पे when the tetranuclear cyst released out, हमने उसको क्या बोला, metacyst बोला, हमने उसको क्या बोला, metacyst बोला. Now these metacysts when undergo mitosis and form 8 daughter nuclei, it is going to form metacystic trophozoite. अब तक क्या था, metacyst था. जैसे ही वो daughter nuclei बन जाता है, daughter cell बन जाता है, we call it as metacystic trophozoite. ठीक है. These young one develop into the feeding stage, क्योंकि क्या है, trophozoite बनेगा तभी तो खाएगा, तभी तो बढ़ेगा, तभी तो डेवलप करेगा, तभी तो मल्टीप्लाई करेगा, राइट. दे इन्वेंट ऑन द म्यूकस मेम्ब्रेन, दैट इज म्यूकोसा एंड सब म्यूकोसा मेम्ब्रेन ऑफ द लार्ज इंटेस्टाइन ग्रो इनटू मैच्योर ट्रोफोजोइट, राइट. सो दिस वाज ऑल अबाउट द लाइफ साइकिल ऑफ एंटामीबा हिस्टोलिटिका. यहाँ पे एक बार देखते हैं क्लास. We'll start with the trophozoite. Trophozoite undergo binary fission and then बहुत बार सी ट्रोफोजोइट है, अंडरगो बाइनरी फिशन के बाद इट फीड्स एंड देन डॉटर सेल्स बनता है, डॉटर सेल्स विल फीडिंग के बाद अगेन बाइनरी फिशन होता है, बाइनरी फिशन के बाद अगेन सेम प्रोसेस, डॉटर सेल्स बनती है, ऐसे बहुत सारे डॉटर सेल्स बन जाएंगे. नाउ दिस इट इज गोइंग टू कहाँ पे हो रहा है, ये वॉल में हो रहा है. दिस इज गोइंग टू एंटर इनटू द ल्यूमेन एंड इट कन्वर्टेड इनटू प्रीसिस्टिक स्टेज. ल्यूमेन में ही व्हेन cyst wall is formed, we call it as cyst stage. cyst stage जैसे ही स्मॉल इंटेस्टाइन में पहुंचता, cyst stage में क्या होता है, आपकी न्यूक्लियस की माइटोसिस होती है. कितनी बार, 2 बार होती है. ड्यू टू दैट व्हाट हैपेंड, सिंगल न्यूक्लियस कन्वर्टेड इनटू टेट्रान्यूक्लियस cyst, सिंगल न्यूक्लियस कन्वर्टेड इनटू द टेट्रान्यूक्लियस cyst. अब ये टेट्रान्यूक्लियस cyst comes out in a fecal matter and can be alive for 10 days. उसके बाद क्या होता है, it reaches the new host through contaminated food and water. Contaminated food and water के बाद अभी जो नए होस्ट में गया है, अगेन सेम एलिमेंट्री किनोल का पाथवे इट इज गोइंग टू फॉलो एंड इट एंटर्स इनटू द स्मॉल इंटेस्टाइन. स्मॉल इंटेस्टाइन में क्या होता है, द सिस्ट वॉल गेट रप्चर बाय ट्रिप्सिन. रप्चर होने के बाद क्या होता है, ये टेट्रान्यूक्लियस सिस्ट कम्स आउट, जिसको हम क्या बोलते हैं, मेटासिस्ट बोलते हैं. एंड रप्चरिंग ऑफ द सिस्ट वॉल को हम क्या बोलते हैं, एक्सिस्टमेंट बोलते हैं, राइट. नाउ दिस मेटासिस्ट व्हाट इट इज गोइंग टू डू, इट इज अंडरगोइंग another mitosis division due to which 8 daughter nuclei is formed. 8 daughter nuclei के अराउंड तुम्हारे साइटोप्लाज्म हो जाते हैं. इन साइटोप्लाज्म के आ जाने की वजह से न्यू डॉटर सेल्स आर प्रोड्यूस, जिसको हम क्या बोलते हैं, मेटासिस्टिक ट्रोफोजोइट बोलते हैं. ठीक है. These metasystic trophozoite are going to become mature trophozoite and again the same cycle will be performed. So this was one cycle. Then what is the second cycle? Let's understand this over here. Okay. Here, see, extraintestinal amoebiasis. It is nothing but sometimes trophozoite may rupture the wall of the capillaries. अब तक हमने क्या देखा था, trophozoite में binary fission होगी, feeding करेगा, rupture करेगा, it converted into pre-cystic stage, cystic stage, and this way में follow होगा, right? But कभी-कभी क्या होता है, it is going to rupture the capillaries, blood vessel को rupture करेगा, and it is going to enter into the bloodstream. Primarily reaches to the liver class. I have already discussed this with you. Always from small intestine, blood always first goes to liver. From liver it goes to other part. Why? Because liver is going to detoxify the things which you have taken, right? So always it goes to liver. तो ये कैपिलरी से सबसे पहले कहाँ जा रहा है, लिवर में जा रहा है. So कैसे हम लिख सकते हैं, let me change the color, ब्राइट कलर नहीं है, ओके ठीक है. ट्रोफोजोइट से कहाँ गया ये, ब्लड कैपिलरीज में कहाँ गया, इट एंटर इनटू द लिवर. लिवर में क्या होता है, इट कॉजेज एब्सिसिस. एब्सिसिस इज नथिंग बट, लेट मी शो यू, इट इज द पस फॉर्मेशन. किधर लिखा, हाँ इधर लिखा. इट इज द कलेक्शन ऑफ पस विद इन द बॉडी. इट इज द कलेक्शन ऑफ पस विद इन द बॉडी. वी कॉल इट एज what do we call it as, abscession. Okay, sorry, I forgot. Abscession. Some may also call it as secondary amoebiasis. Now from there they may go to lungs. तो अब यहाँ से liver में it is going to cause abscession. आप इस तरीके से फ्लो चार्ट बना सकते हैं. abscesses के बाद वहाँ कहाँ जाएगा, lungs में जाएगा, lungs से heart में जाएगा, फिर आपका kidney में जाएगा. Then it goes to gonads. What are gonads? Gonads are your reproductive organ. And it causes abscession. That means collection of pus. हर जगह आपकी pus collection होने लग जाएगी, which is the severe pathological condition. That's what we have seen over here. Some become mature trophozoite and enter into extraintestinal. Extraintestinal means apart from elementary canal. Okay, so extraintestinal मतलब क्या पढ़ा हमने? Intestine से blood capillaries में आ जा रहा है. Blood capillaries से it goes to liver. Liver से it can go to lung, it can go to kidney, it can go to gonad. And क्या form करता है? Abscesses form करता है. That is nothing but the collection of pus, right? So this cycle is super important class. You are supposed to draw in your notebook. Now, this whole life cycle we have written in one page. See here, trophozoite in large intestine undergoes binary fission, produces number of daughter Entamoeba which feed on the bacteria and host tissue. Trophozoite grow in size, again undergo binary fission, and it increases in number. Some trophozoite enter into the lumen of the large intestine. When it is entering into the lumen, we call it as pre-cystic stage. Now, when it develops the cyst wall around itself, we call it as cystic stage. Okay, this process we call it as Excystation. Excystation we call it as, spelling I'm forgetting, just write it out. Okay, Excystation. Now enter into the cystic stage. After that, 2 mitosis division takes place and 4 daughter nuclei are produced, and this 4 daughter nuclei overall we call it as tetranuclear cyst, which is basically the infective stage, and it comes out through the fecal matter. Let me erase it. It comes out through the fecal matter and stay alive for 10 days. Now let's see, it is the infective stage. That means इस stage में वो human के अंदर आता है. तो through contaminated food and water, it is going to reach the new host and reaches to the small intestine. There, trypsin की help से cyst wall rupture होती है. Due to rupture of cyst wall, what will happen? Tetranuclear cyst release out. And जब ये release हो जाता है, now we call it as metacyst. Metacyst में 1 mitotic division होती है. Already यहाँ पे 4 nucleus से 1 mitotic division से कितने? 8 daughter nuclei बन गए. All these 8 daughter nuclei are surrounded by the cytoplasm. We call it as metacystic trophozoite. Okay, जो daughter cells produce होते हैं उसको हम metacystic trophozoite बोलते हैं. And it started feeding, and feeding करने के लिए वो develop कर जाता है किसमें? Feeding stage है ना, मतलब किसमें develop करेगा? It develops into the trophozoite. Again I have repeated this. Now, after trophozoite, same process, it enters into the mucus, that means mucosa and submucosa, and grows into mature trophozoite. Again, same cycle follows over here. Okay, so this was all about the life cycle of Entamoeba histolytica. Super important with respect to your board. Now, let's jump on the pathogenicity. Pathogenicity is nothing but pathogenic. That means pathogen हमने क्या पढ़ा था? Organism which causes disease. तो वो किस तरीके से Entamoeba histolytica is pathogenic to us? How it is causing disease on us? So trophozoite क्या release करता है? Histolysin we have already studied, right? It is going to break your tissue. Now trophozoite goes deep into submucosa and it causes ulcer. तो क्या होता है यहाँ पे? If it goes into deep into mucosa, तो छाले जैसे पड़ जाते हैं. जैसे अगर आपका mucosa is not present on the stomach, तो क्या होगा? आपका HCl आपके सेल को इन्फेक्ट करेगा और वहां पे छाले जैसे हो जाएंगे, लीसन्स बन जाएंगे. उसको हम क्या बोलते हैं? अल्सर बोलते हैं, राइट? दीज अल्सर कंटेन सेलुलर डेब्रीज. क्योंकि क्या हुआ? यहाँ का सेल आपका डिस्ट्रॉय हुआ ना, तो यहाँ का सेल डेब्रीज प्रेजेंट होगा, टुकड़े प्रेजेंट होंगे. डेब्रीज मतलब टुकड़े प्रेजेंट होंगे. लिम्फोसाइट इज नथिंग बट योर WBC. Lymphocyte is nothing but your WBC. And then blood corpuscles, that means other components of the blood will be present and bacteria will be present. It leads to the formation of abscission. इसकी वजह से क्या होता है? There is a collection of pus. कहाँ पे? Wall of the large intestine में. Ultimately stool में आपका क्या आएगा? Blood and mucosa आएगा. Mucus आएगा, sorry. Okay, mucosa तो लेयर का नाम है. Mucus चिपचिपा सा substance होता है जिसको हम mucus बोलते हैं. This condition we call it as amoeboid dysentery or intestinal amoebiasis or tropical amoebiasis. Repeating myself again, trophozoite है, उसके पास enzyme है histolysin. It is going to infect the submucosa layer and उसकी वजह से अल्सर कॉज होता है. अल्सर अगर हम बात करें तो what does it contain? It contains the cellular debris, WBC, blood corpuscles and bacteria. इसकी वजह से क्या होता है? Abscessus कॉज होता है, that is collection of the pus. और उसकी वजह से क्या होता है? आपके large intestine में stool में आपका blood and mucus present होता है. और इस सारे phenomenon को जो blood में जो लूज मोशन होता है आपका, जिसमें ब्लड भी रहता है, म्यूकस भी रहता है, उस तरीके के लूज मोशन को हम क्या बोलते हैं? एमोबॉइड डाइसेंट्री और इंटेस्टिनल एमियोबाइसिस और ट्रॉपिकल एमियोबाइसिस. Then some person do not exhibit any symptoms. Such kind of person we call it as asymptomatic or carrier. जैसे हमने कोरोना टाइम पे पढ़ा था कि बहुत लोगों को कोरोना था बट हमें डिटेक्ट नहीं हो पाता था, तो they were asymptomatic, उनको कोई symptoms नहीं आते थे. They were asymptomatic and they act like a carrier. Their stool contain tetranucleus cyst and spreads parasite to the other person. Why they are called as carrier? क्योंकि उनके स्टूल में टेट्रान्यूक्लियस सिस्ट तो है, वो स्प्रेड तो हो रहा है, इट्स जस्ट दैट उनको कोई सिम्टम्स नहीं मिल रहा है. दे आर बीइंग इफेक्टेड, दे एक्ट लाइक अ कैरियर, बट इट्स जस्ट दैट हमें पता नहीं चलता है कि दे आर हैविंग दैट डिजीज. नेक्स्ट वन, प्रोफाइलैक्सिस. इट इज नथिंग बट व्हाट आर द प्रिवेंटिव मेजर्स व्हिच यू कैन टेक. First one is you're using boil and filter water, washing fruits and vegetable properly, because ये कहाँ से आ रहा है, contaminated food and water में आ रहा है ना. Now washing hands before eating and using septic tank toilets. So that's all we have in Entamoeba histolytica life cycle. Please go through the life cycle once again. It is super important with respect to the board. Thank you.